Enzyme catalysis is the increase in the rate of a chemical reaction by the active site of a protein. The protein catalyst may be part of a multi-subunit complex, and/or may transiently or permanently associate with a cofactor. Catalysis of biochemical reactions in the cell is vital due to the very low reaction rates of the uncatalyzed reactions. A key driver of protein evolution is the optimization of such catalytic activities via protein dynamics. The mechanism of enzyme catalysis is similar in principle to other types of chemical catalysis. By providing an alternative reaction route, the enzyme reduces the energy required to reach the highest energy transition state of the reaction. The reduction of activation energy increases the amount of reactant molecules that achieve a sufficient level of energy, such that they reach the activation energy and form the product. As with other catalysts, the enzyme is not consumed during the reaction but is recycled such that a single enzyme performs many rounds of catalysis. Induced fit. The favored model for the enzyme-substrate interaction is the induced fit model. This model proposes that the initial interaction between enzyme and substrate is relatively weak, but that these weak interactions rapidly induce conformational changes in the enzyme that strengthen binding. The advantages of the induced fit mechanism arise due to the stabilizing effect of strong enzyme binding. There are two different mechanisms of substrate binding: uniform binding, which has strong substrate binding, and differential binding, which has strong transition state binding. The stabilizing effect of uniform binding increases both substrate and transition state binding affinity, while differential binding increases only transition state binding affinity. Both are used by enzymes and have been evolutionarily chosen to minimize the activation energy of the reaction. Enzymes that are saturated, that is, have a high affinity substrate binding, require differential binding to reduce the energy of activation, whereas small substrate unbound enzymes may use either differential or uniform binding. These effects have led to most proteins using the differential binding mechanism to reduce the energy of activation, so most substrates have high affinity for the enzyme while in the transition state. Differential binding is carried out by the induced fit mechanism. The substrate first binds weakly, then the enzyme changes conformation, increasing the affinity to the transition state and stabilizing it, so reducing the activation energy to reach it. It is important to clarify, however, that the induced fit concept cannot be used to rationalize catalysis. That is, the chemical catalysis is defined as the reduction of I euro relative to I euro in the uncatalyzed reaction in water. The induced fit only suggests that the barrier is lower in the closed form of the enzyme but does not tell us what the reason for the barrier reduction is. Induced fit may be beneficial to the fidelity of molecular recognition in the presence of competition and noise via the conformational proofreading mechanism. Mechanisms of an alternative reaction route. These conformational changes also bring catalytic residues in the active site close to the chemical bonds in the substrate that will be altered in the reaction. After binding takes place, one or more mechanisms of catalysis lowers the energy of the reaction's transition state, by providing an alternative chemical pathway for the reaction. There are 6 possible mechanisms of over-the-barrier catalysis as well as a through-the-barrier mechanism. Proximity and orientation equals. This increases the rate of the reaction as enzyme-substrate interactions align reactive chemical groups and hold them close together. This reduces the entropy of the reactants and thus makes reactions such as ligations or addition reactions more favorable. There is a reduction in the overall loss of entropy when 2 reactants become a single product. This effect is analogous to an effective increase in concentration of the reagents. The binding of the reagents to the enzyme gives the reaction intramolecular character, which gives a massive rate increase. However, the situation might be more complex, since modern computational studies have established that traditional examples of proximity effects cannot be related directly to enzyme entropic effects. Also, the original entropic proposal has been found to largely overestimate the contribution of orientation entropy to catalysis. Proton donors or acceptors equals. Proton donors and acceptors, that is acids and base, may donate and accept protons in order to stabilize developing charges in the transition state. This typically has the effect of activating nucleophile and electrophile groups, or stabilizing leaving groups. Histidine is often the residue involved in these acid-base reactions, since it has a pKa close to neutral pH and can therefore both accept and donate protons. Many reaction mechanisms involving acid-base catalysis assume a substantially altered pKa. This alteration of pKa is possible through the local environment of the residue. pKa can also be influenced significantly by the surrounding environment, to the extent that residues which are basic in solution may act as proton donors, and vice versa. It is important to clarify that the modification of the P-core unregistered trademark S is a pure part of the electrostatic mechanism. Furthermore, the catalytic effect of the above example is mainly associated with the reduction of the pKa of the oxyanion and the increase in the pKa of the histidine, while the proton transfer from the serine to the histidine is not catalyzed significantly, since it is not the rate-determining barrier. Electrostatic catalysis stabilization of charged transition states can also be by residues in the active site forming ionic bonds with the intermediate. These bonds can either come from acidic or basic side chains found on amino acids such as lysine, arginine, aspartic acid or glutamic acid, or come from metal cofactors such as zinc. Metal ions are particularly effective and can reduce the pKa of water enough to make it an effective nucleophile. Systematic computer simulation studies established that electrostatic effects give, by far, the largest contribution to catalysis. In particular, it has been found that enzyme provides an environment which is more polar than water, and that the ionic transition states are stabilized by fixed dipoles. This is very different from transition state stabilization in water, where the water molecules must pay with reorganization energy in order to stabilize ionic and charged states. Thus, the catalysis is associated with the fact that the enzyme polar groups are reorganized. The magnitude of the electrostatic field exerted by an enzyme's active site has been shown to be highly correlated with the enzyme's catalytic rate enhancement. Binding of substrate usually excludes water from the active site, thereby lowering the local dielectric constant to that of an organic solvent. This strengthens the electrostatic interactions between the charged polar substrates and the active sites. In addition, studies have shown that the charge distributions about the active sites are arranged so as to stabilize the transition states of the catalyzed reactions. In several enzymes, these charge distributions apparently serve to guide polar substrates toward their binding sites so that the rates of these enzymatic reactions are greater than their apparent diffusion control limits. Covalent catalysis Covalent catalysis involves the substrate forming a transient covalent bond with residues in the enzyme active site or with a cofactor. This adds an additional covalent intermediate to the reaction, and helps to reduce the energy of later transition states of the reaction. The covalent bond must, at a later stage in the reaction, be broken to regenerate the enzyme. This mechanism is utilized by the catalytic triad of enzymes such as proteases like chymotrypsin and trypsin, where an acyl enzyme intermediate is formed. An alternative mechanism is Schiff base formation using the freon from a lysine residue, as seen in the enzyme uglase during glycolysis. Some enzymes utilize non-amino acid cofactors such as pyridoxal phosphate or thiamine pyrophosphate to form covalent intermediates with reactant molecules. Such covalent intermediates function to reduce the energy of later transition states, similar to how covalent intermediates formed with active site amino acid residues allow stabilization, but the capabilities of cofactors allow enzymes to carry out reactions that amino acid site residues alone could not. Enzymes utilizing such cofactors include the PLP-dependent enzyme aspartate transaminase and the TPP-dependent enzyme pyruvate dehydrogenase. Rather than lowering the activation energy for a reaction pathway, covalent catalysis provides an alternative pathway for the reaction and so is distinct from true catalysis. For example, the energetics of the covalent bond to the serine molecule in chymotrypsin should be compared to the well-understood covalent bond to the nucleophile in the uncatalyzed solution reaction. A true proposal of a covalent catalysis would require, for example, a partial covalent bond to the transition state by an enzyme group, and such effects do not contribute significantly to catalysis. Bond strain equals, this is the principal effect of induced fit binding, where the affinity of the enzyme to the transition state is greater than to the substrate itself. This induces structural rearrangements which strain substrate bonds into a position closer to the conformation of the transition state, so lowering the energy difference between the substrate and transition state and helping catalyze the reaction. However, the strain effect is, in fact, a ground state destabilization effect, rather than transition state stabilization effect. Furthermore, enzymes are very flexible and they cannot apply large strain effect. In addition to bond strain in the substrate, bond strain may also be induced within the enzyme itself to activate residues in the active site. Quantum tunneling equals, these traditional over-the-barrier mechanisms have been challenged in some cases by models and observations of through-the-barrier mechanisms. Some enzymes operate with kinetics which are faster than what would be predicted by the classical Igar-Euro. In through-the-barrier models, a proton or an electron can tunnel through activation barriers. Quantum tunneling for protons has been observed in tryptamine oxidation by aromatic dehydrogenase. Interestingly, quantum tunneling does not appear to provide a major catalytic advantage, since the tunneling contributions are similar in the catalyzed and the uncatalyzed reactions in solution. However, the tunneling contribution is likely crucial to the viability of biological organisms. This emphasizes the general importance of tunneling reactions in biology. In 1971 to 1972 the first quantum mechanical model of enzyme catalysis was formulated. Active enzyme equals, the binding energy of the enzyme substrate complex cannot be considered as an external energy which is necessary for the substrate activation. The enzyme of high energy content may firstly transfer some specific energetic group X1 from catalytic site of the enzyme to the final place of the first bound reactant, then another group X2 from the second bound reactant must be transferred to active site to finish substrate conversion to product and enzyme regeneration. We can present the whole enzymatic reaction as a two-coupling reactions. S1 plus EX1 equals S1 EX1 equals P1 plus EP2, S2 plus EP2 equals S2 EP2 equals P2 plus EX2. It may be seen from reaction that the group X1 of the active enzyme appears in the product due to possibility of the exchange reaction inside enzyme to avoid both electrostatic inhibition and repulsion of atoms. So we represent the active enzyme as a powerful reactant of the enzymatic reaction. The reaction shows incomplete conversion of the substrate because its group X2 remains inside enzyme. This approach as idea had formerly proposed relying on the hypothetical extremely high enzymatic conversions. The crucial point for the Vr cation of the present approach is that the catalyst must be a complex of the enzyme with the transfer group of the reaction. This chemical aspect is supported by the well-studied mechanisms of the several enzymatic reactions. Let us consider the reaction of peptide bond hydrolysis catalyzed by a pure protein I plus or minus chymotrypsin, which is a well-studied member of the serine proteases family, C. We present the experimental results for this reaction as two chemical steps. S1 plus EH equals P1 plus EP2, EP2 plus H equals EH plus P2. Where S1 is a polypeptide, P1 and P2 are products. The IRST chemical step includes the formation of a covalent acyl enzyme intermediate. The second step is the desilation step. It is important to note that the group H+, initially found on the enzyme, but not in water, appears in the product before the step of hydrolysis, therefore it may be considered as an additional group of the enzymatic reaction. Thus, the reaction shows that the enzyme acts as a powerful reactant of the reaction. According to the proposed concept, the H transport from the enzyme promotes the IRST reactant conversion, breakdown of the IRST initial chemical bond. The step of hydrolysis leads to a breakdown of the second chemical bond and regeneration of the enzyme. The proposed chemical mechanism does not depend on the concentration of the substrates or products in the medium. However, a shift in their concentration mainly causes free energy changes in the IRST and enal steps of the reactions, and due to the changes in the free energy content of every molecule, whether S or P, in water solution. This approach is in accordance with the following mechanism of muscle contraction. The enal step of ATP hydrolysis and skeletal muscle is the product release caused by the association of myosin heads with actin. The closing of the actin binding cleft during the association reaction is structurally coupled with the opening of the nucleotide binding pocket on the myosin active site. Notably, the enal steps of ATP hydrolysis include the fast release of phosphate and the slow release of ADP. The release of a phosphate anion from bound ADP anion into water solution may be considered as an exergonic reaction because the phosphate anion has low molecular mass. Thus, we arrive at the conclusion that the primary release of the inorganic phosphate H2PO4 leads to transformation of a significant part of the free energy of ATP hydrolysis into the kinetic energy of the solvated phosphate, producing active streaming. This assumption of a local mechanochemical transduction is in accord with Terosha Euro unregistered trademark S mechanism of muscle contraction, where the muscle force derives from an integrated action of active streaming created by ATP hydrolyses. Examples of catalytic mechanisms: in reality, most enzyme mechanisms involve a combination of several different types of catalysis. Triose phosphate isomerase Triose phosphate isomerase catalyzes the reversible interconversion of the two triose phosphates isomers dihydroxyacetone phosphate and E-glyceraldehyde 3-phosphate. Trypsin Trypsin is a serine protease that cleaves protein substrates after lysine or arginine residues using the catalytic triad to perform covalent catalysis, and an oxyanion hole to stabilize charge buildup on the transition states. Aldolase Aldolase catalyzes the breakdown of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Enzyme diffusivity. The advent of single-molecule studies led in the 2010s to the observation that the movement of untethered enzymes increases with increasing substrate concentration and increasing reaction enthalpy. Subsequent observations suggest that this increase in diffusivity is driven by transient displacement of the enzyme's center of mass, resulting in a recoil effect that propels the enzyme. Reaction similarity. Similarity between enzymatic reactions can be calculated by using bond changes, reaction centers or substructure metrics. See also, catalytic triad, enzyme assay, enzyme kinetics, protein dynamics, quantum tunneling, the proteolysis map, time-resolved crystallography, enzyme promiscuity. References. Further reading, Allen Fersht, Structure and Mechanism in Protein Science, A Guide to Enzyme Catalysis and Protein Folding. W. H. Freeman, 1998. ISBN 0-7167-3268-8, dedicated issue of Philosophical Transactions Beyond Quantum Catalysis in Enzymes freely available.